All right, I'm going to pick up with the third installment of our chapter 1 notes, or chapter 16 notes, part 1, excuse me, with males. And this is where I left off. So we've talked about the anatomy of the testes, we've talked about the anatomy of the duct system, we've talked about the 3 accessory glands, which sperm does not actually flow through itself, but they're responsible for producing seminal fluid. So let's talk about semen. So what is in semen? So semen contains, let me get a little cursor. Semen contains sperm, and it also contains the fluids created by the accessory glands. And sperm cannot survive without seminal fluid. Again, in that fluid, it has energy, so glucose, fructose, so that it can produce ATP. It has vitamins that are essential. It has substances that are there to activate sperm and increase their motility. And it also has mucus that is responsible for neutralizing the environment of the urethra on semen's way out, as well as neutralizing the pH of the vagina. As you can see, seminal fluid has a pH of about 7.26 to 7.6, so it is alkaline, and that is on purpose because sperm motility actually declines as acidity increases. And so the alkaline pH of semen is there to neutralize the vagina. But again, semen is a transport medium for sperm, provides nutrients, and neutralizes. So the world is not going to end, or the species is not going to end due to lack of sperm, let's just say. In a sperm producing male, under normal hormone conditions, about 400 million new sperm are produced in a man per day. That's pretty crazy. So 400 million new cells per day in the testes and the seminiferous tubules. Sperm can be produced by males from puberty throughout a male's entire lifetime, unless there are hormonal issues, or for some reason that male loses his testes. So again, sperm production begins at puberty, but the whole concept of sperm production actually begins during fetal development. And sperm start off as sperm stem cells that are called spermatogonia. And during fetal development, testosterone that is produced during fetal development causes spermatogonia to be produced. And basically, spermatogonia are just going through mitosis. And so we know that mitosis is the creation of 2 identical daughter cells. So during fetal development and during childhood in boys, spermatogonia are just simply going through rapid mitotic divisions, so that we're getting more and more and more and more fresh, new, just born sperm stem cells. At puberty, FSH, which comes from the anterior pituitary hormone or follicle stimulating hormone, is produced. It goes up, okay, and it causes the stem cells to become 2 different types. They either become type A or type B spermatogonia. So again, this rise in FSH at puberty causes spermatogonia, which have just been basically non-differentiated and just dividing and dividing and dividing up to this point, causes them to become type A or type B. Type A stem cells are going to continue to divide and just kind of keep the sperm stem cell population going forever. Type B cells are actually going to now start to push towards the lumen. So this kind of all occurs in the outer periphery of the seminiferous tubules. So again, if I were going to draw the seminiferous tubules, so we've got these A cells that are just going to keep going through mitosis out here. And then we have B cells that I like to consider they're type B, so that means that they're up to bat. And they're going to start the process of spermatogenesis, and they're going to move eventually and end up in the lumen of the seminiferous tubules. So once a stem cell has become a B stem cell and it's going to move through the process of spermatogenesis, we now call them primary spermatocytes. So they're just the beginning. So spermatocytes, or, not quite sperm cells yet. The primary spermatocytes are diploid. That means that they have 46 chromosomes. And if you have taken biology before, you know that our gametes, so our egg and our sperm cells, have 23 chromosomes. They are haploid. So the process of spermatogenesis is not actually mitosis, but it is mitosis's more complicated sister, meiosis. And essentially, meiosis is 2 rounds of mitosis, but with only 1 DNA replication. And this will yield to half the amount of genetic material in the daughter cells, if that makes sense. So in spermatogenesis, we go from diploid primary spermatocytes to haploid spermatids. Spermiogenesis, and I want you to focus on this part right here. Spermiogenesis is the process of sperm maturing. So spermatogenesis is going to become a sperm. Spermiogenesis is going from immature to mature sperm, and that occurs in the epididymis. This whole process of production to maturation is going to take about 64 to 72 days. So again, here we have a spermatogonium, and this would be a B type. So a type B spermatogonium. And it's going to move, so like over here, like this would be the wall of the seminiferous tubules, and this would be the lumen, okay? So the B spermatogonium are now going to become here, and they're going to become primary, do some math symbols, primary spermatocytes. They're going to go through meiosis 1. After meiosis 1, we are going to call them secondary spermatocytes. And they both have 46 chromosomes, but they have 23 sister chromatids together. So they essentially have double copies of the same thing bound together. And then after meiosis 2, or right before meiosis 2, something kind of crazy happens. And these sister chromatids actually will switch alleles. They'll kind of trade their Pokemon cards with each other. Because as you know, when people have, your siblings, if you were not born, as an identical twin, all of these sperm that end up being produced can create very, genetically different appearing people by way of, their phenotypical features, eye color, gender. And that's because of this thing that happens right before meiosis 2 begins. So when our secondary spermatocytes are created, they're not considered to be fully diploid. They're haploid, but they do have 46 chromosomes. They have double the amount of DNA. Again, they start swapping alleles. There's this trading that happens. And then as the secondary spermatocytes, go into meiosis 2, they end up creating 4 haploid spermatids that are not identical. So this might show it a little better. So here we have, you can either have 2 types of spermatogonium. You can have A or B. Essentially, the A would be the ones that keep going through mitosis, And then it's supposed to be an A. I'm sorry, I'm writing with my finger on my mouse. Oh, I could do this. There we go. And then B are down here. Oh, I finally figured out that my computer's touch screen. Amazing. And then the B spermatogonium become the primary spermatocytes. So the primary spermatocytes are 2N, they are diploid, and they're going to go through meiosis 1. In meiosis 1, all right, again, there are 46 chromosomes, but there are 23 pairs of sister chromatids together. And those sisters, again, trade their Pokemon cards, and they're like, well, I'm going to give you a blue eye for and you take the brown eye, and I'm going to take tall, and you take short, and they switch these alleles with each other. And then at the end, these secondary spermatocytes are going to turn into 4 haploid spermatids. And again, if you look at those spermatids, they don't look like our traditional sperm. They will then be pushed into the lumen of the seminiferous tubules and then move on through the reedy testis and the epididymis to become the haploid sperm that we know. So this is the wall of the seminiferous tubule, okay? And so this would be more superficial and this would be more deep. And then here's the lumen right there, if that makes sense. Let me erase my stuff here. I'm going to erase 2. There we go. So these would be the spermatogonia out here. So these are the diploid cells. The A stem cells would keep going through mitosis. Eventually, A stem cells are going to become B stem cells, and it's just going to be as they, get pushed down towards the lumen. Okay. When you have a B stem cell become a primary spermatocyte, again, it will go through meiosis, which has 2 phases, meiosis 1 and meiosis 2, with the end result becoming 4 haploid, not identical daughter cells. Okay, so here we have the primary spermatocyte going through meiosis 1. Here's the secondary spermatocyte, which will then go through meiosis 2, which results in the 4 haploid, which means they have 23 chromosomes, non-identical cells. And as you can see down here, the sperm are going to get produced and then pushed into the lumen, and then they will go to the reedy testis. This right here is a Sertoli cell. And I told you that we had 2 different types of interstitial cells in the testes. We had Leydig cells and Sertoli cells. Sertoli cells, I'm writing with my finger now, not sure if that's better. Those are called nurse cells. And they are responsible for providing nutrients, support, and protection to the developing spermatocytes and spermatogonia. And then we have Leydig cells that are in the testes, and they actually produce testosterone. That's what T2 stands for, is testosterone. And then the last thing I'm going to talk about is the anatomy of sperm. So mature sperm have 3 distinct regions. They have what's considered a head, a midpiece, and a tail. There is also a specialized little helmet called an acrosome. So in the head, that is basically where all 23 chromosomes sit. And I like to joke that males' sperm are just like typical males in packing. They have a pair of underwear, a toothbrush, maybe an extra shirt. Okay. Sperm, they're responsible for delivering those 23 chromosomes to the egg, and that's it. That's their only job. Okay. They're not developing, an embryo or a fetus. They just have to say, here's my 23 chromosomes. Egg, do it. Okay. Eggs are totally different. They have lots of excess cytoplasm, but sperm need to be these little streamlined swimming missiles. Okay. So in the head, we have 23 chromosomes packed. In the midpiece right there, you're going to see mitochondria that are there to, make ATP to help, aid in motility,